I'm Myla T. Sarmiento, and now I discuss the difference of translation from transcription. Transcription. Transcription is the first step in gene expression. It involves copying a gene's DNA sequence to make an RNA molecule. Transcription is the process of copying out the DNA sequence of a gene in the similar alphabet of RNA. The main enzyme involved in transcription is RNA polymers. RNA polymers, which uses a single-stranded DNA template to synthesize a complementary strand of RNA. Specifically, RNA polymers build an RNA strand in the 3 to 5 direction, adding each new nucleotide to the 3 end of the strand. There are 3 steps of transcription: the annotation, elongation, and termination. Step 1. Annotation. Annotation is the beginning of transcription. It occurs when the enzyme RNA polymers bend to the region of a gene called promoter. This signals that the DNA to unwind the enzyme can read the bases and one of the DNA strand. The enzyme is now ready to make a strand of mRNA with a complementary sequence of bases. Step 2. The elongation. Elongation is the addition of nucleotides to the mRNA strand. RNA polymers read the unwound DNA strand and build the mRNA molecules. Using complementary base pair, there is a brief time during this process when the newly formed RNA is bound to the unwound DNA. During this process, an adenine in the DNA bends to the uracil U and the RNA. Step 3. Termination. Termination is the ending of the transcription and occurs when RNA polymers crosses a step termination sequence in the gene. The mRNA strand is complete and decays from DNA. And also there are all stages of transcription. Transcription of a gene takes place in 3 stages: the annotation, elongation, and termination. Here we will briefly see how this step happen in bacteria. You can learn more about the details of each stage and about how eukaryotic transcription is different in the stages of transcription article. The first stage is the annotation. RNA polymers binds to the sequence of the DNA called the promoter, found near the beginning of a gene. Each gene or group of the transcribed genes in bacteria has its own promoter. Once bound, RNA polymer separates the DNA strand, providing the single-stranded templates needed for transcription. The promoter region comes before and slightly overlaps with the transcribed region whose transcription is specified. It contains recognition site for RNA polymers or its helper protein to bind to. The DNA opens up in the promoter region so that RNA polymers can begin transcription. The elongation. One strand of DNA, the template strand, acts as template for RNA polymers. At its read these templates one base at a time. The polymers build an RNA molecules out of complementary nucleotides, making a chain that grows from 5 to 3. The RNA transcript carries the same information as the non-templates or coding strand of DNA, but it contains the base uracil or U. Instead of thymine or T, RNA polymers synthesize an RNA transcript complementary to the DNA template strand and in the 5 to 3 direction. It moved forward along the template strand in the 5 to 3 direction, opening the DNA double helix as it goes. The synthesized RNA only remains bound to the templates around template strand for a short while, then exits the polymers as a dangling string, allowing the DNA to close back up and form a double helix. Stage 3. The termination. Sequence called terminator signal that the RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymers. An example of termination mechanism involving formation of hairpin in the RNA is showing below. And this is the example. The terminator DNA encodes a region of RNA that form a hairpin structure followed by a string of U or nucleotides. The hairpin structure in the transcript causes the RNA polymers to stall. The U or nucleotides that comes after hairpin for weak bonds with a nucleotides of the DNA templates, allowing the transcript to separate from the template and ending transcription. There are 3 main steps to the process of DNA transcription. First is annotation. DNA is transcribed by the enzyme called RNA polymers. Specific nucleotide sequence tell RNA polymers where to begin and where to end. RNA polymers attaches to the DNA at the specific area called the promoter region. The DNA and promoter region contains a specific sequence that allow RNA polymers to bind to the DNA. Second is the elongation. A certain enzyme called transcription factor unwind the DNA strand and allow RNA polymers to transcribe only a single strand of DNA into a single-stranded RNA polymers called messenger or RNA. The strand that serves as the template is called the antasized strand. The strand that is not transcribed is called the sense strand. Like DNA, RNA is composed of the nucleotide bases. RNA, however, contains the nucleotides adenine, guanine, cytosine, and uracil. When RNA polymers transcribe the DNA, guanine pairs with cytosine, or G over C, and adenine pairs with uracil, A or A and U. The third is termination. RNA polymers move along the DNA until it reaches a terminator sequence. At that point, RNA polymers release the mRNA polymers that they catch from the DNA. Translation. Take a moment to look at your hand. The bone, skin, and muscle you see are made up of cells, and each of those cells contain many millions of proteins. As a matter of fact, proteins are key molecular building blocks for every organism on Earth. Ever wonder how antibiotics kill bacteria? For instance, when you have a sinus infection, different antibiotics work in different ways, but some attacks a very basic process in bacterial cells. They knock out the ability to make new proteins. There are 3 steps of translation. Your cells are making new protein every second of the day, and each of those proteins must contain the right set of amino acids linked together in just the right order. That may sound like a challenging task, but luckily your cell, along with those of other animals, plant and bacteria are up to the job. To see how cells make protein, let's divide translation into 3 stages. The annotation starting off, elongation adding onto the protein chain, and termination finishing up. First step, the annotation. In annotation, the ribosome assembles around the mRNA to be read into first tRNA, carrying the amino acid methionine, which matches the start codon, or AUG. This setup, called the Annotation Complex, is needed in order to translation to get started. In order for translation to start, we need a few key ingredients. These include a ribosome, which comes in 2 pieces, large and small, an mRNA, with instruction for the protein will build, an initiator, tRNA carrying the first amino acid in the protein, which is almost always methionine. Second step is the elongation. Elongation is the stage where the amino acid chains get longer. In elongation, the mRNA is read 1 codon at a time, and the amino acid matching each codon is added to a growing protein chain. But how does the chain actually grow? To find out, let's take a look at the first round of elongation after the annotation complex has formed, but before any amino acid have been linked to make a chain. Termination, the third step. Termination is the stage in which the finished polypeptide chain and release. It begins when a stop codon enter the ribosome, triggering a series of events that separate the chain from its tRNA and allow it to drift out of the ribosome. After termination, the polypeptide may still need to fold into the right 3D shape, undergo processing such as the removal of amino acids, give shift to the right place in the cell, or the combine with other polytypes before it can do its job as a function protein. And that's